I welcome you all to my science cafe. For a change, I'm going to be selling you apples in my cafe, and later during the talk, I'll give you some caffeine as well. So how many of you have a relative who has diabetes? I have my father, my mother, my brother, all have type 2 diabetes, and I'm constantly worried because diabetes increases the risk of heart failure. There are so many factors behind this link, but the mechanisms are not very clear. And in our lab, we are studying one such mechanism. First of all, let's see how glucose is taken into the heart. So here I have a cell that is a cardiomyocyte or a heart cell from a rat. So on the surface of the cell, we have insulin receptors. When insulin binds onto this receptor, it initiates a series of signaling cascade that leads to expression of these transporter proteins on the surface, and glucose is taken inside. And this glucose goes to the mitochondria, and mitochondria produces energy for the cell. So heart constantly pumps 100,000 times per day. So it needs a continuous supply of energy. And in terms of type 2 diabetes, what happens? This signaling cascade doesn't work. So these glucose transporters do not go to the cell surface. So glucose cannot get into the cell. So it accumulates within the blood. So we constantly see an increased blood glucose level. However, our body tries to bring in glucose during type 2 diabetes itself by arranging another alternative transporter. So that is called sodium glucose link transporter. So during diabetes, our body increases this transporter protein. However, while it brings glucose, it also brings 2 sodium ions within the cell. So the sodium concentration within the cell increases, so the positive charge increases. So let us see what are all the negative effects of having more sodium within the cell. So here, assume this is a heart cell, and I want you to focus on 3 important components. One is this plasma membrane. It should have a negativity inside and positivity outside so that it maintains a membrane potential. Another one is mitochondria, the powerhouse, which continuously creates ATP that is necessary for the contractile machinery to continuously contract and relax. And another one is sarcoplasmic reticulum, which is a calcium store. So when calcium is released by these calcium releasing channels called ryanodine receptors, excitation and contraction occurs. Here, this cardiomyocyte contracts and relaxes, and then when you measure the calcium, then you can see that it goes up and down. So it's contraction and relaxation phase or systole and diastole phase. During diabetes, what happens is due to increased expression of this protein, sodium concentration within the cell increases, and this affects the membrane. Membrane gets depolarized, and also it reduces the mitochondrial calcium. And this results in increased reactive oxygen species like peroxides. This can damage the calcium releasing channels of the sarcoplasmic reticulum. So instead of releasing just calcium during the systole, it can also release calcium during the diastole. So all these can lead to arrhythmia. Arrhythmia is irregularity in beats. So we hypothesize that if we block this transporter protein, then we can reduce the sodium and stabilize the membrane and reduce the calcium leak during diastole and reduce arrhythmia. So for this, we are using fluorescein. This is a polyphenolic compound naturally found in apples, bark, and trees, and also in the fruits. So here we use a diabetic animal model, and we harvest the heart from the rat and isolate the cardiomyocytes. And the first experiment, we loaded the cells with some sodium indicator and then incubated the cells with fluorescein. So these are like pure compounds from the apple trees and barks. And when we measured the sodium concentration inside the cell, when we treated the cells with fluorescein, the concentration went down. So this proved our first hypothesis. And next, we measured the diastolic calcium leak. So this is a classical experiment. We stimulated these cells externally by field stimulation and then rested the cells. So if we find calcium leak during this phase, then there's going to be arrhythmia. So here you can see the transients, and then at the end we apply caffeine, which totally opens all the calcium channels within the sarcoplasmic reticulum and floods the cytoplasm with calcium. And here in this phase, we can observe the calcium release, spontaneously released calcium as sparks. All these white spots indicate the calcium sparks. And if these white spots are more, the animal is going to have arrhythmia. So when we measured this calcium spark frequency compared to the control rats, the hearts from the diabetic rats had higher frequency of these sparks. And when we incubated the cells with fluorescein, the spark frequency went down. So this proved our second hypothesis that fluorescein can reduce the spark frequency or the diastolic calcium leak. And in the third experiment, we measured the membrane potential. So as this transporter is electrogenic, it can bring more positive charge into the cell and depolarize the membrane. So in the diabetic rats, the cell had less negative charge. So this difference can be very meager, but it can mean a lot in terms of excitability. So these diabetic cells are very excitable, leading to arrhythmia. And then when we incubated the cells with fluorescein, again, this depolarization was like reversed, and it was significantly different compared to the diabetic rats. So the membrane potential was stabilized by fluorescein. And then we finally wanted to see if it works on the heart itself. So we harvested the heart and perfused continuously with the buffer containing oxygen, glucose, and calcium, and then hooked some ECG electrodes and measured ECG directly. And here you can see the normal rhythm here. And then we perfused the heart with caffeine and isoprotrinol. So caffeine contracts the heart, it increases the force of contraction, and the other drug increases the rate of contraction of the heart. So when we challenge the heart like this, a normal heart will withstand this challenge, but if the heart is a little compromised, then it will show arrhythmia or irregular beats. So we measured the duration of arrhythmia, like there'll be episodes of arrhythmia like ventricular tachycardia. And then we also counted individual arrhythmic events. So when we treated the heart with fluorescein, the duration of arrhythmia as well as the individual arrhythmic events went down. So this proved our fourth hypothesis. So this is the summary showing that by inhibiting this transporter protein with fluorescein, we can reduce the sodium concentration, stabilize the membrane potential, and reduce the diastolic calcium leak and reduce arrhythmia. So our study paves the way for identifying more drug targets that can specifically target this protein on the heart as a treatment for reducing the incidence of heart failure in diabetes. And also our studies tells the importance of the sodium ion within the cell, contributing to heart failure in diabetes. With this, I thank you all for listening, and my PI, Dr. Sandra Despa, and my current and former lab members. Thank you very much.